The next technique that we're going to look at is the spread plate technique, which is a quantitative technique. Here, the primary goal is the enumeration of viable aerobic microorganisms, which is reported as a CFU, meaning a colony forming unit. The principle is that you place a known volume of a diluted sample on the air surface and you spread it evenly. And after incubation, each colony represents one viable cell or cell cluster from that plate of dilution. The procedure is that you first do the serial dilutions of your liquid sample and then you pip around 0.1 milliliter onto the pre-solidified agar plate. You spread it evenly using a sterile L-shaped spreader, as you can see on the slide right there, and you incubate the plates in an inverted fashion in order to avoid the condensation dripping onto the surface. A key detail in this procedure is the countable range. You aim for around 30 to 300 CFUs per plate, and below that the random error becomes too large, and above that the colonies merge and the morphology isn't necessarily decipherable, so you can't count accurately. So the countable range must be in between the values of 30 and approximately 300. The spread plate technique is commonly used for CFU calculations, and CFU calculations are applicable in food, water, and pharmaceutical microbiology, and because the colony morphology is very, very clear and the colonies only grow on the surface. A couple limitations regarding the spread plate technique is that it is restricted to aerobic microorganisms and it requires serial dilutions, and there's a limited sample volume per plate, and this is because you're typically just spreading only a small volume of around 0.1 milliliters.